problem with the grading? Because I think
Oh, yeah, the focus on it, right?
yeah, because I think there's I think everything was graded. I think some things were graded incorrectly. Some people, all the sections I heard got like zeros or 30s, and I wouldn't stress over it. All right, so today we're going to start and finish the urinary system. Next week we'll start and finish the reproductive system, review, and in 2 weeks is your last test for lecture. Okay? Lots to do, very little time to do it. All right, so we're going to see that the urinary system comprised of 2 kidneys, okay? Left and right. Coming from the kidneys, we have a tube called the ureter, which empty down into our bladder and exit through a urethra. We can see anatomically, when we open up the kidney, very similar to the adrenal gland. Okay, so as we know, the adrenal gland sits atop both of the kidneys, so we're going to see that it'll have an outer capsule, it'll have a cortex, and it will have a medulla. Some things that the kidneys are responsible for to control your body's homeostasis of body fluids and its ions. So we'll see that the urinary system is responsible for doing that. It helps also regulate your body's pH. It'll help regulate blood volume, blood pressure. It produces specific hormones. Okay, remember when we talked about hypovolemic shock, we kind of left out that last pathway this way. Okay? But the kidneys also help in reducing our blood volume or increasing blood volume. Okay? It helps regulate your blood glucose, and importantly also excretes our waste products. Okay, here's what the kidney looks like. It's about the size of our fist. So it's really not that large, but yet it's a very powerful organ. Here we can see, just basically on a transverse section, how posteriorly the kidneys are. Okay? They're really right back on that posterior abdominal wall. Okay? All behind the intestines. So this is going to help us for Tuesday. We'll go through this anatomy again in the lab, but this will help right now with some of the structures that we have to go over. So if we open up the kidney, okay, we're going to see a couple of structures. One, we have an outer capsule. Okay, so here's our outer capsule of the kidney. Cortex is this very light tan structure that goes all the way around. So we have a renal cortex. And then we also have a renal medulla. So everything all in here will be medulla. Okay? Just like with the adrenal gland, you have your cortex with the different zones. You have the medulla. Okay? Once again, if the adrenal gland sits atop here, it makes sense. Anatomically, it's going to be very similar because they come from the same embryonic tissues. A couple other structures. We have these pyramid-like structures that we can see inside the kidney, hence we call them our renal pyramids. The tip of the pyramid has a specific name. We call that the renal papilla. Okay? So we have our pyramid. We have the renal papilla. Between the pyramids, we have some tan tissue. Okay, those are called columns. So what separates the pyramids are renal columns. If we take a piece like this, so we have cortex, medulla, pyramid, papilla, some column, we call that a renal lobe. So you'll have a lobe here, you'll have another lobe here, 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 and here. Not only can we break it into a pyramid, cortex, medulla, we can break it into what we call a renal lobe. Okay? We have other structures. At the tip of the papilla, eventually when urine drains from the kidney, we have what's called the minor calyx. Okay? So at every papilla, so here, 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 and here, we have a structure called the minor calyx. The minor calyx come together and group into what we call the major calyx. So all in here, all in here. And then eventually the major calyx drains into a portion called the renal pelvis, right in here. In the area of the kidney there, just like with the lungs, we call that the hilum. So just like the hilum of the lung, we have the hilum of the kidney. Okay? And it's not the ileum. It's the hilum.
No.
So we're going to have our renal artery, we'll have our renal vein, renal pelvis, we'll have the renal nerve, and we'll have lymphatic vessels as well, very similar to the lungs. Okay, we had our blood supply instead of a pelvis, we had the primary bronchi, but we also had lymphatics and nerve. From the hilum, we come down into our ureter. The ureter is a tube that's going to drain all the urine out of the kidney, all the way down into the bladder. Okay? Think of the ureter like the esophagus. It has to connect two portions. So the ureter is just a muscular tube, passageway. Okay? So being muscular, once again, it can do a little bit of dilations and constrictions. It does a little peristaltic movement with the fluid that's inside, and it drains down into your bladder. Now the opening of the ureter, okay, is only a few millimeters in diameter, so you're not talking very, very wide. Okay? If it's as wide as a straw, that's going to be as big as you get. So when somebody has kidney stones, okay, try sucking out a grape through that straw. It's just not happening. Okay? So we'll talk about kidney stones later in the class. The functional unit of our kidney is what's called a nephron. The nephron is where we're going to make urine. Right, so we're going to go over different parts of the nephron. We have about 1 million of them in each of our kidneys. Okay. However many we are born with is however many we keep. Okay. You can live without one of your kidneys, so you can at least filter your blood enough with one kidney. However, you're not going to be at optimization. Okay? 2 kidneys give you 100% filtration of your blood. 1 kidney is going to give you about 80%. So it'll do its job, it's just not going to be as good as having 2. But you can survive with just 1. We're going to look in a couple minutes that, let's say somebody had an adrenal gland tumor. Okay, and they needed to get the adrenal gland taken out. They may take away some tissue from the kidney. Okay? If we go back a second, okay? Here's our adrenal gland. It may be impossible to just take the gland without taking a portion of the kidney, especially if there are some cells that have gone into your kidney, okay? Metastasized. But because of the blood supply that we have and how it circulates, we're going to see that you can actually cut away a piece of your kidney and it will still be a functioning organ. Okay? Because the way the blood is intertwined amongst our kidney, it's very possible just to take away a portion of it, whether it's the top or the bottom, and it will still function and filter. Eventually, when the nephron makes urine, okay, urine needs to drain through a whole bunch of structures. So it goes through a collecting duct, then eventually down into the minor calyx, major calyx, then it goes into the renal pelvis, ureter before it reaches the bladder, to eliminate it through the urethra. Okay? So it has to go through here to here, all through here, and eventually drain down into our bladder. Okay? Here's a kidney taken out of a cadaver. Okay? If you were to look at a kidney inside a person, it should be pink in nature, like a lung, pink tissue. It's healthy. Okay? If it's got darkened vessels, obviously something disease is happening in this kidney, but it should be pink in nature. Once again, maybe as big as our fist, so it's really not that big, but yet does a lot of important things for our body. Okay. So let's take a look at some of the blood supply to the kidney. So once again, we have our renal artery bringing in oxygenated blood. And the renal artery is going to divide. We'll get what's called a segmental artery. From our segmental artery, we're just going to keep dividing. We'll get what's called the interlobar artery. Okay, remember the lobe? Okay? So this is an interlobar artery. At what I call the base of the pyramid. So if here's the tip, here's the base. From interlobar artery, we get what's called our arcuate arteries. From our arcuate arteries, we get what's called interlobular arteries. I think your lab manual actually has them named cortical radiate arteries. So it can go by two names. Okay, these interlobular, these small ones, can either be interlobular arteries or cortical radiate arteries. Okay? From there, we're going to be getting to the blood supply that's around our nephron. Okay? So we're going to skip here for a second. Okay? So we have our renal artery giving way to segmental arteries. Segmental arteries giving way to interlobar arteries. Interlobar arteries giving way to arcuate arteries. Arcuate arteries giving way to interlobular. And then here's where we are around our nephron. Okay? Think back to when we talked about the alveoli. What surrounds an alveola? What surrounds that cluster of grapes? Capillaries, right? So you can do gas exchange. The kidney isn't going to be doing gas exchange, but it's going to be doing different molecule exchanges. Okay? We're going to be filtering your blood. The kidney's a big filtering organ. We're going to be filtering out things that we don't want, but yet keeping things we do want. So it's a big filter, so we need the blood to be in close proximity. From our interlobular arteries, we go to afferent arterioles. Okay, that's an arteriole that's going to respond to our body's pressure. Whether we have increased blood pressure, decreased blood pressure. It's going to give way to a structure called the glomerulus. So we'll have glomerular capillaries. We'll show you all these in a second. Then eventually out through efferent arterioles. And then depending on the type of nephron that we have, and we're going to go through the anatomy of these two different types, we either have peritubular capillaries, or this could be the vasa recta. So right here, this will change depending on the type of nephron that we have. Okay? So it would either be efferent arterioles giving way to peritubular capillaries, or efferent capillaries giving way to the vasa recta. And then we get into the venous side, because now we have deoxygenated blood. So now that blood's going to go back into where? Right? Renal vein into what? Renal vein hooks into what? Vessel. Remember? What's the big vein in our belly? Say it. The inferior vena cava. Okay. See? And then that's going to go back up to the heart. Okay. All right, so the two different types of nephrons that we have are either cortical nephrons or juxtamedullary nephrons. Okay? Cortical nephrons or juxtamedullary. We have two different types. We're going to go through the difference between the two. This is the cortical one. That's the juxtamedullary nephron. They look similar, but this one is more in the cortex of the kidney, and this one has a long loop in the medulla of the kidney. Okay? So if we look at all the structure that's in yellow, okay? Whatever's in yellow is the nephron. Everything in red is capillaries. Okay? So it starts off with this structure right here. Okay? It looks like a C. Okay? We call that Bowman's capsule or the glomerular capsule. Bowman's capsule or the glomerular capsule. Off of Bowman's capsule, we get a bunch of twisting and winding little tubes. These tubes actually twist and wind in all the different planes of our body. So some will go sagittally, some will go transverse, some will go coronally. And because they're tiny little tubes, we call them tubules. So the first set of twisting and winding tubes we call the proximal convoluted tubule. Proximal because it's closest to Bowman's capsule, convoluted because they twist and turn, and tubule because it's a tiny little tube. Okay? Almost like a little capillary tube. Okay? Sometimes we refer to this as the PCT, proximal convoluted tubule. A lot of times in the notes or in your lab out, you might see PCT. And you can abbreviate as long as you don't interchange the letters. Off of the proximal convoluted tubule, we get this big loop. Right? This is called the loop of Henle. The loop of Henle has a descending limb and an ascending limb. And we're going to see that depending on whether we're talking about the descending limb or the ascending limb, it does different things. Okay? Off of the ascending limb, we get a second set of twisting and winding little tubes. That we call the distal convoluted tubule. It's further away, it's twisting and winding, and there are little tubes. So our distal convoluted tubule, or our DCT. That is what makes up our nephron. Bowman's capsule, PCT, the descending and ascending limb of the loop of Henle, and our DCT. Inside the nephron, there's fluid. Right? This fluid is not yet urine. So it can only be called tubular fluid. Okay? Your body is not yet finished filtering it out. So we can't call it urine. It can only be called tubular fluid. What connects the nephron to our collecting duct, okay, is just a connecting piece called a connecting tubule. So each nephron will be connected to our collecting duct. Okay? So there might be another nephron here, here, another one here. Okay? So it connects to a connecting tubule into what we call the collecting duct. This long tube here, that is your collecting duct. In the collecting duct, we're going to have a hormone that will fine-tune the tubular fluid. It comes from our posterior pituitary. Anybody want to give a guess what it is? ADH. Okay? ADH is going to be responsible for doing one last refinement of our tubular fluid, because eventually that fluid will reach the papillary duct, because here's the renal papilla, and then down here, as the renal papilla drains into the minor calyx, that's when we can finally call it urine. Okay? So it's not yet urine until we're draining into our minor calyx. Okay? So any fluid in here is just tubular fluid. and then eventually now we have our urine. Okay? And that's where it'll drain from the minor calyx into the major calyx, from the major calyx into the renal pelvis down into the ureter to the bladder. Okay? Everybody good so far? Okay. Now if we look around this entire cortical nephron, you see how the capillaries are in close proximity? Okay. Just like in an alveolus, we have the capillaries and lymphatic vessels in close proximity. What your body does is when blood comes into the kidney, everything that's in your blood gets poured into the nephron. All of your glucose, all of your amino acids, your sodium, your calcium, your chlorine, everything. Everything that's in your blood gets poured into the nephron. Then your body says, wait, hold the fort. I need some of those things. I can't let you get rid of them, because what do we need glucose for? What's our main thing that we need glucose for? And what production? Remember from AP1? Cellular what? What's the main energy that we produce? ATP. So cellular metabolism, we need glucose, right? Don't we start with glucose? Right? And then we go through our Krebs cycle, electron transport chain, and then we make our ATP. So we can't urinate it out, we need it. So as things go from our blood into the nephron, your body says, wait, I want to take it back. We call that reabsorption. Right? When we talk about reabsorption in the urinary system, it means we're taking things and putting them back into your bloodstream. We're reabsorbing it back. We're taking it back. So we're going to see that when everything from the blood gets dumped into the Bowman's capsule, as it travels through the PCT, we're taking some things back into our blood. As the fluid goes through the descending and ascending limb, we're taking things back into the blood. In the DCT, same thing. And then more refinement in here. So initially, we give it up, but we're going to see through the process of glomerular filtration and through going through the rest of the nephron that we're going to take things back. Now if we look inside Bowman's capsule, okay, right here is a capillary structure called the glomerulus. Okay? The glomerulus is just a bunch of capillaries that are intertwined. If we look at the glomerulus and Bowman's capsule together, okay, so these two structures, that's what makes up a renal corpuscle. So if we're talking about the renal corpuscle, we're talking about Bowman's capsule, or the glomerular capsule, and the glomerulus. Okay? So the beginning part of the nephron and this capillary structure right here. Okay? If we look back to where the glomerulus begins, okay, here's our pyramid. So this is renal medulla. Here's cortex, right? The lighter tan tissue. Okay. This is a cortical nephron because most of the nephron is in the cortex. Okay. Here are the arcuate vessels, arcuate artery, arcuate vein. Okay. This vessel here. Okay. Remember, that could be the interlobular artery or that cortical radiate artery. Okay? Either one. Off of that interlobular artery, we have that afferent arteriole. Okay? Remember, we had our list of vessels, and we had our renal artery, segmental artery, interlobar artery, arcuate artery, interlobular, then we went into that afferent arteriole. Yes? Okay. So now we're right here. Okay. The afferent arteriole is what's allowing blood to come in and supply the glomerulus. As blood leaves the glomerulus, it exits through your efferent arteriole. Okay? Here's the afferent, here's the glomerulus, here's the efferent arteriole. Then we said, depending on the type of nephron, whether it was a cortical nephron, we have our peritubular capillaries, which are all of these. Those are your peritubular, peri means surrounding, tubular capillaries. And then we get into the venous structure, okay? Now here are the venous structures. Okay? The interlobular veins will drain back into the arcuate veins, which will drain back into the interlobar veins, which drains back into the renal vein, which drains back into your vena cava, back up to the heart. Okay? If we look for a second at the juxtamedullary nephron, it has the same structures. So it has Bowman's capsule, it has PCT, it has loop of Henle, it has our DCT. The only difference is the really, really long loop of Henle, which extends deep into the medulla. So that's how we call it juxtamedullary nephron, because of the really long loop of Henle. Okay. Otherwise, it has to connect through a connecting tubule, collecting duct, capillary duct, and eventually urine drains into the minor calyx. They're going to be doing the same thing. It's just location within the kidney will determine which nephron you're going to see. And in actuality, as you open up a kidney, you cannot see any nephrons. Okay? If we look at this kidney, can you see nephrons? No. They're microscopic. Okay? They have to be. We have 1 million of them in each of our kidneys. Okay? All we can see are the minor calyx into the major calyx into the renal pelvis. Everybody good so far? Yeah. Now the kidney, like I said, is responsible for filtration. Think back a second when we talked about blood vessels. Okay? And we talked about capillaries. What type of capillaries do we have? We have contiguous capillaries, we have fenestrated capillaries, and we have sinusoidal capillaries. Right? Somewhat ringing a bell? Okay. These are capillaries. Okay? They're covered by cells. The cells have little appendages to them. Let's get a bigger picture. Here we go. Okay? The cell is called a podocyte. This helps create the outer covering of the capillary. Podocytes have little fingers. Between the fingers, there are openings. Those were those slits that we talked about when we were talking about the fenestrated capillaries. So we can have different size particles coming in and out of the blood and through the different pores of the inner lining. Okay? So if you think of the palm of your hand as a podocyte, here's the little finger appendages, okay? Here and here. This would be another podocyte. So you put your fingers like this, there's spaces between it. So the space is the filtration slit, and the palm of your hand is the podocyte. Okay? We're going to see that on the model in lab. Now, when everything from the blood comes in through the afferent arteriole and into the glomerulus, there are pressures that occur which cause everything to be dumped into this little space. Okay? So everything that's in your blood will be ejected out into that space. Because of the pressure that we have in our body forcing things out. The space, okay, is called capsular space. Bowman's capsule, call it capsular space. So just think of it as putting your hand in a mitten. Okay, if your hand and fingers are the glomerulus, the mitten is Bowman's capsule. There's some space between your hand and the glove, right? That's the capsular space. The pressure that's coming in through the afferent arteriole is called your glomerular blood hydrostatic pressure. Okay, the pressure that comes in is called your glomerular blood hydrostatic pressure. Okay. We have a blood pressure, right? Okay. So being that this is a smaller artery, right? An arteriole is just a smaller artery. It has a pressure. Right? All our arteries have some type of a pressure, whether it's a big aorta or a radial artery, it has a pressure. Okay? Obviously, the pressure is not as great as the aorta, right? Because otherwise, these would burst. Okay? So by the time we get down into an arteriole and then a capillary, right, pressures have dropped. But we can't measure the pressure. So normally, your glomerular blood hydrostatic pressure, or GVHP, is around 55 millimeters of mercury. Everybody's a little different depending on your own blood pressure. That's the pressure that's coming in this way. Okay? Opposing that are two different pressures. Okay? You have a pressure inside the glomerulus, and you have pressure that's inside capsular space. They're going to be pushing up against that GVHP. The pressure inside the capsule is called your capsular hydrostatic pressure. It is a measured value of 15 millimeters of mercury. The pressure inside the glomerulus, the blood colloid osmotic pressure, measures 30 millimeters of mercury. Okay? So you have one pressure coming in, while two pushing up against it. Okay? Look at the numbers. Which one is higher? Okay? The first one, going in this way. So therefore, if we take 55 minus 15 minus 30, we have a net of 10. Right? We call that the net filtration pressure. Okay? Your net filtration pressure. As long as your net filtration pressure is a positive number, things can exit your blood and be dumped into the capsular space. Everybody understand that? Okay. As long as it's a positive number. So that means there's enough pressure coming in to oppose the two that are pushing up against it. Okay. So that means anything in your blood can be dumped into this capsular space of Bowman's capsule. Right?
So the difference, so you said as long as it's
A positive number. As long as it's a positive number, whether it's 1 or whether it's 21, that's a positive number, right? That means anything in your blood can be dumped into this space. If it's a negative number, things cannot exit your blood. So that means your body will not be filtering. Okay? If it's a negative number, you don't want that. If your net filtration pressure is -1, -5, -10, your kidneys will not filter your blood. Okay, so that's not a good thing. Okay? We want it to stay around 10. That's its homeostatic number. Okay? If the pressure starts to go higher, that net filtration pressure starts to get to like 15, 20 or even 30, things are going to be starting to be pushed through the nephron way too fast. Your body's not going to be able to filter the way it should. Okay? So your body's homeostatic NFP should stay about here. Now, what are some things your body's doing? Well, if we look, once everything is in Bowman's capsule, okay, we said everything in the blood gets dumped into it. We can't have that. Okay? We can't get rid of our body's glucose. We can't get rid of our body's amino acids. We can't get rid of our ions. We need them. Okay? You need your calcium. You need your potassium. You need your chlorine. Okay? So we're going to take stuff back. Remember we call that reabsorption? Okay? Reabsorption. In the PCT, okay? So this first set of winding tubes, we're going to take back all of the glucose that we dumped into the capsular space. That's why those capillaries have to be in close proximity to the nephron. Okay? We're going to take it back. We want it. Your body says, I can't get rid of it. I need it. We're going to take back all of our amino acids. Okay? What do our amino acids give us? They make our what? Our proteins. We need that. Okay? In the PCT, okay, we're going to absorb or reabsorb 65% of our water, sodium, potassium, 50% of our chlorine. Okay? But yet, we're not going to take back hydrogen ions, ammonia, urea, because we're going to eliminate those. We don't want to take those back, so they'll stay in that tubular fluid. As fluid goes into the loop of Henle, we're reabsorbing more water, more sodium, more potassium, calcium, and magnesium. Urea is still going to stay in there. We don't want to get that out of that tubule. That's going to be waste. Okay? In our DCT, okay, here's what we're reabsorbing. And then eventually, ADH comes into play. And ADH, once again, anti-diuretic. Okay? That's going to give us, okay, either dilute or concentrated urine. How do we know if it's dilute or concentrated? By color. Okay? We can look. If urine is very dark in color, very amber in color, it's concentrated. If it's clear, it's dilute. It's like opening up a can of frozen orange juice. Okay? It's concentrated, it's dark, we dilute it with water. Okay? So in the morning, most likely your first urine is concentrated, okay? Because your body wants to conserve its water. You haven't been drinking for 3 hours, 4 hours, 8 hours, depending on how much you sleep. Okay? But as your day goes by, you're taking in fluid, you're taking in more water. It starts to get more clear, okay? Because you're hydrating. Okay? Did we lose anybody yet? Yes? No? Where? Where did we lose you? Because this is the only day we're going to have for urinary. Come on, where did we lose you? Okay? So, in the corpuscle, once again, this is going to be the first place that your body starts to do some type of filtering. Remember, the corpuscle is Bowman's capsule and the glomerulus. Right? Bowman's capsule and glomerulus. So everything, once again, gets dumped into the capsular space. So we call that glomerular filtration. That's the beginning part of how our body's going to make our urine how we begin filtering our blood. So again, initially, because of the pressure coming in is greater than the two pressures pushing up against it, anything and everything in your blood is dumped into that capsule. As we go through the rest of the nephron, okay, once again, look how close proximity the capillaries are to the nephron. We're either going to reabsorb things back into the blood, called tubular reabsorption, okay, or we're going to secrete things out back into the tubule. Secrete because we don't want it. We're going to give it up because we don't need it. It'll be waste. And then eventually, by the time we get into our collecting duct and ADH does its last minute fine tuning, we'll have concentrated or diluted fluid, which we can now call urine. Okay. On the opposite side, okay, of Bowman's capsule, we have what we call the DCT. Remember the distal convoluted tubule. Further away, it twists and winds. Now inside the DCT, there are specialized cells. These specialized cells are called the macula densa cells. The macula densa cells, along with your afferent arteriole, okay, so this vessel and these cells make up what's called the JG apparatus, juxtaglomerular apparatus. Okay. Since these are two separate structures, how do they communicate? Okay, one's a blood vessel, and one's a tube that has fluid. Even though they're side by side, how are they going to communicate? Between the two structures, okay, there are specialized cells called mesangial cells. Okay? So figure between two students, okay, there's something to help them communicate. So it's taking up the space between them. So what's taking up the space between these two vessels are cells. The afferent arteriole and the macula densa cells of the DCT. DCT, not the PCT. They make up the JG apparatus. Here is where, in response to pressure changes in our body, your body gives off hormones. And your kidney gives off a hormone. That way the afferent arteriole can either constrict or dilate, and it can change the pressure either coming into the kidney. So if a vessel constricts, pressure is going to go up, right? Or if we constrict the vessel, we get increased blood pressure. If we make it bigger, it drops. So this is why the kidney, okay, because of these two structures, responds to blood pressure changes in our body. And hormones get released, okay, from our blood vessel into the DCT. Okay. We create what's called renin and angiotensin. When we finally get down to where we have our urine, remember the ureters, which we have from one of each kidney, drains into a big muscular vessel called your your bladder. Okay? The only purpose of your bladder is to hold the urine. Okay? So as it fills up, it expands. Very similarly to the stomach. Because on the inside we have rugae, just like the stomach has rugae. Those structures enable both of those organs to get bigger. Surrounding our bladder, we have muscle. We call it the detrusor muscle. As the detrusor muscle starts to stretch, because our bladder is filling up with urine, we have what are called stretch receptors. Stretch receptors, once your bladder fills up to a certain amount, trigger off what we call the Micturition reflex. That's what tells you, all right, my bladder is full enough, I need to go pee. Okay? So as the bladder gets bigger and bigger, muscle gets stretched, okay? And just like with the lungs, when they start to expand enough, we had a reflex, okay, called what? Which said don't overinflate. What was the reflex? Brewer reflex. Okay, we have enough air inside, don't overexpand, so I don't want to pop like a balloon, and then we can squash out all our air. Okay? Your bladder is never gonna burst, because eventually you'll have to urinate. You may be able to hold it for a while, but eventually you're going to have to urinate. Okay? If we look at the bottom part of the bladder, we have two sphincters. Okay? Just like in the digestive system, a sphincter is just muscular constrictions. Okay? So we have an internal sphincter, which is involuntary, and we have an external sphincter, which is voluntary. So, initially as children, we can't hold fluid in our bladder because we've yet, have learned to control our external urethral sphincter. Okay, it's a muscle, so it has to be trained and get stronger. But as we get older, okay, and we learn to hold our urine, we're strengthening this sphincter, we're strengthening this muscle. And then eventually, we get older, we get tired, we get lazy, our muscle atrophies, and we can't hold our urine anymore. Okay? So we start out in diapers, and we end up in diapers. Okay?
You really can't burst your bladder? Ever?
No.
Well, sometimes when a patient may take the full meal and they can't urinate after like the 8 hours they give them to urinate.
There could be inflammation in the tissue, which is blocking the urethra, or something that's blocking it, or could aggravate urine exiting out. Because that's a foreign tube getting stuck in a small opening, and then you're pulling it out. So sometimes when you do that, don't get, it's the inner lining is very smooth. So if you stick something in and now you pull it out, you now scrape that inner lining, which creates inflammation. When you have inflammation, it's going to narrow the opening, so nothing can exit. So you have to be careful, you have to monitor, because people can't.
So those sensors that you were talking about on the inside, when saying the urine is building up.
The stretch receptors are in the muscle walls. It's in the muscle. It's like in the heart, how we had the conduction system, the SA node, AV node. Okay, they're embedded in the muscle tissue. The stretch receptors are in the muscle. You're not gonna open up the bladder and say, oh, here's a stretch receptor. Here, there's one here, there's one there. Okay. Now, if we look anatomically where our bladder is located, okay, here's the bladder in females, Okay? You have one urethra. Okay? If you look where the bladder is located, the uterus sits atop. So as you go later on in your pregnancy and the uterus is getting bigger and bigger and bigger, it's not letting your bladder expand. So therefore, as you're drinking more water for a purpose to maybe stop contractions and stay hydrated, your bladder can't hold as much water as if you weren't pregnant because it can't expand. So you constantly have to urinate. Because look anatomically where the uterus is and located to the bladder. In the male system, okay, we only have our bladder. We don't have anything sitting atop, okay? But we have two different urethras. Okay? So different a little bit anatomically. But right now we're just going to be concentrating on the bladder. When we get into the reproductive system, we'll talk about the rest of the anatomy, and the same thing with the female system. We need to take a couple minutes break. So I can't talk anymore. All right, so take a 10-minute break, and we'll come back.
¿Cómo se queda?